Thank you, and thank you everyone for joining us tonight. I'm just always happy to have people learn about my work and also share their questions about it and maybe their own ideas of where to take it forward. So yeah, I'm going to be telling you about this method that I built and some of its results. It's a method for barcoding cell types with color, so that in a living animal, so in vivo basically, we can tell exactly what cell type is what. In this case, these are neurons, and I'll tell you about that in a second, and I'll get into more details pretty soon as well. But the idea here is that we can look at dynamics of what take place in these cells as a result. And here, this is actually a worm coiled around itself, and this is its tail here, and its head is here, and you can already see the technique on display. These colored balls here, they're neurons. They're actually the nuclei of neurons. And they all have unique colors so that we can tell their identity, but all the animals look the same. And so in every animal, we're going to have this little yellow cell here, for example, this yellow neuron. And I can tell you exactly what that neuron is because of its coloring. It's in this case, it's a neuron that is called AVD. So the neurons usually have 3 to 4 letter names, and then there's a code to let you know that they're on the left or right side of the animal. All that stuff we'll get into in a second. But for every one of these colors that you see here, I can tell you what neuron you're looking at. Doesn't matter how old or young the animal is, or whether it's a male or a female. And so I'll get into that right now. Let me go forward here. First, just to tell you a little bit about my organism, since I think a lot of people might not be familiar with it. It's C. elegans. It's these very small nematodes. Here you can see one crawling around a dish. So this uses some work from my PhD, in which a camera is just following this worm around here. It's backing up. Its head is right about here, and its tail is here. And as an adult, they're about 1 millimeter long, so real tiny, totally harmless. They just live on petri dishes and eat bacteria all day long. But the cool thing is that they have 80% genetic homology to humans, which means that a lot of the genes that we have, they have as well, and we can study those genes within them, and they have a very compact genome as well, which means that if we knock out a gene, usually there's nothing else to substitute for it. And so we see an effect. We know what that gene is. There's no other genes that kind of, uphold or basically correct for the phenotype, as there would be basically in other organisms. And they go through 4 larval stages to adulthood, and that takes about 3 days. So in 3 days, we've got another 300 to 400 more. They start out as an egg, as you see here, and they hatch into this first larval stage, which we call L1. About 12 hours later, they move to L2, and so on and so forth until they reach adulthood, in which case they're usually a hermaphrodite, which can inseminate herself. But ultimately, the hermaphrodite is a somatic female, so we often refer to them as females. And if the population is stressed, we get a bunch of males as well to kind of help the animal sexually reproduce and spread those genes, mix up the genome a little bit. And if they're under severe stress, so normally, by the way, they would live about 3 weeks, but under severe stress, they go into this alternative life stage called dower, and there they can last for months on end. And basically, these are long-lived animals, and the sad but surprising thing is that these went up in I think it was the Challenger, the Columbia shuttle, and that shuttle exploded. But these are the only thing to survive from that explosion, right? So these fell back to Earth and were still alive by some miracle. So they're pretty hardy. And I'm particularly interested in the worm's nervous system. The adult hermaphrodite has 302 neurons, and the wiring is known. We call that the connectome. So we know the synaptic connectivity between all these neurons now throughout a variety of different ages. That's been built in through recent work. And so we know how this nervous system is wired. And those neurons all have names that are about 3 to 5 letter codes. For example, AWCL, which stands for Amphid Wing Cell Type C on the left side of the worm, which is one particular neuron here. So that all sets up the background for my work. And so first I'll tell you about the big picture itself. My own interest is in the systems biology of the nervous system. And what I mean by that is I want to understand the roles of these individual neurons and then how they all come together and holistically output behaviors. But the bottleneck that we face in this field and in other organisms as well, is neuronal ID. And so what I mean by that is, for example, here on the left side, you'll see this picture. This is the head of a worm. This is its pharynx here. And what you're seeing in white is the expression of a receptor, a GABA receptor, on the surface of a variety of neurons, right? So you can see this GABA receptor is in a lot of neurons. If we wanted to understand the role of this receptor in communication, we'd want to understand which neurons those are, but we have no identity to them, right? We just see a bunch of neurons in place here. And so if we knew their identity, we'd be able to bring more into play in terms of understanding this nervous system. Similarly, here on the right is again the head of a worm. And so these white things here very much correspond to the neurons you're seeing on the left. They're flashing. This worm has GCAMP in its neurons. It's a calcium sensor, and that ultimately is a sensor of neuronal activity. And so these flashing lights are neurons that are active. Again, you see a bunch flashing, but we have no idea which ones those are. So we don't know the roles of individual neurons in the activities you see here, which ultimately leads to behavior. And we don't understand how they then communicate together to output that behavior. But if we had the identity of these neurons, then we'd be able to kind of fill in a lot of the details of how this nervous system operates. And so what I did is I built a multi-purpose tool for neuronal ID, one that would allow me to identify the gene expression you just saw there in the head of the worm, the neuronal activity that you also just saw in the previous slide, and even identify cell fate characteristics. And I'll get back to that, and that'll all make sense as I proceed through the talk. And the solution I came up with was using colored barcodes to ID neurons. So coming back to that picture I just showed you of a GABA receptor and a variety of neurons, right? We have no idea which neurons these are that this GABA receptor is expressed on the surface of. This is a GFP reporter for that particular GABA receptor. We bring in the NeuroPAL, which is the method I built. It's a neuronal polychromatic atlas of landmarks. And this is what it looks like. So here, these are, once again, neurons, and these little colored dots are neurons. These are their nuclei, actually, these colored dots. They have the same color across all worms. So this is not a rainbow method. It's very, very different. It's not stochastic in that sense. It's entirely deterministic. Every worm here basically looks identical. We have one color map for them, and those colors stay the same from birth till the death of the worm, as far as we know. I haven't really looked at a worm right on its deathbed, but I've never seen these colors differ. And in both sexes, and even through this dower stage, for those who are more familiar with the different worm life stages. And because of that, I can then combine this this reporter here for a GABA receptor in white with the NeuroPAL and tell you every single neuron that has that GABA receptor expressed on its surface. So that sets up everything for the talk itself. And then the first thing I just want to tell you about is the existing cell labeling methods and where this kind of fits in with them. So I think the one that people are most familiar with is Brainbow. It's a stochastic method of coloring different cells, in this case, neurons. The problem here, though, is that it's entirely random what coloring you get. So from one animal to the next, the same neuron type will receive very different coloring. And even within the same animal, the same type of neurons will always receive random coloring, so they'll just look different. You can't tell the neuron type as a result. There are these sequential staining methods like Clarity and Marefish, which are quite popular now. And the problem here is that it takes multiple rounds of staining to determine the contents, the inventory of particular cells, and then determine the particular cell type. And the other problem really is that this can only be done in fixed tissue, in dead animals, right? So we can't look at any dynamics in that sense. I mean, we could maybe piece together, but it's a lot of work. But we're basically dealing with a dead animal, so we can't look at neuronal activity, for example, with this technique. And then the last one was a really nice method that was created called Clades right around the time when I was working on my method. It's a means of coloring. Oh, is there a question? Maybe not. Okay. It's a means of coloring cells by their lineage, but we only have 5 colors here, and so we can only color 5 different cell types. And so the challenge I was facing was basically a method of building, getting stereotype coloring so that we could very quickly determine the different cell types that we're looking at in vivo, so in a living animal. And the challenge in particular for me, because I'm interested in the nervous system, And I'm interested in the entire nervous system. I'm interested in the systems biology of the nervous system, a holistic view of it. It was one strain in which we could ID every single worm neuron. So to get into that, this is an overview, real quick overview of the worm and its 302 neurons in the adult hermaphrodite. This is its head right here. This is its mid-body, and the vulva is right here. The eggs come out of here. And this is the tail right here. And this, I just want to, as an example, basically focus on one particular ganglion, which is the retrovesicular ganglion, which is located right here. The name doesn't matter, and even the neuron names aren't going to matter, but it really just is meant to exemplify the problem that we're facing here. These are electron micrograph reconstructions from 3 adult worms. So this is not my work. This work was done in the 80s, and we've had it for a very long time. These are 3 adult hermaphrodites, roughly around the same age. And this is the retrovesicular ganglion, which contains about 20 neurons. And here we have the ground truth. So we had basically slices of the worms reconstructed to know where the neurons are. And then I've basically circled, or in the electron micrographs, basically circled where the nuclei are in the rough size. And what I want you to focus on is these 2 neurons here, DB1 and DA1. Name doesn't matter, but I just want to show you that in the first adult hermaphrodite, they are right next to each other. And in the second adult hermaphrodite, they found in the 80s, that they are separated by a neuron. There's a neuron between them here. And in the third adult hermaphrodite, there are 3 neurons between them, and they've switched their orientation. So now DA1 is the anterior neuron and DB1 is the posterior neuron. The key takeaway here is that the position, even though we talk about the worm, the stereotype of the worm's nervous system, and yes, all adult hermaphrodites have 302 neurons, in the same 302 neurons. And when we get to the level of the actual ganglia, we see a distinct lack of stereotypy. We see basically that the positions alone are not sufficient to ID the neurons. They kind of shuffle around, and where they're necessarily positioned in one animal to the next is not clear unless we bring in the coloring. So I think the next question I want to address is, well, I just told you that there's some positional variability that takes place at the level of the ganglia. How extreme is it? And that was one of the first questions we wanted to address. What you're seeing here is a cartoon that was drawn for one of the worms that was reconstructed in the 80s. In green is its pharynx, so the food comes in through here. This is the head of the worm, and here you can see a variety of neurons in a particular ganglion. So this is a lateral ganglion. Here it's one of the ganglia that contains neurons. It's a sac of neurons. The same neurons all the same, all the time in these sacs, but where they're positioned in the sacs, we see some movement. And so what we did is we took 10 NeuroPAL worms and we identified the neurons across them, or I did, and thus determined where their mean position is. And that's what this dot is here. So this diagram that you're seeing below here, this lines up exactly with the cartoon you're seeing there. So this is the army dorsal neuron. Name doesn't matter again, but this neuron here you're seeing in the cartoon is the same neuron down here, and same for this neuron here. These arrows kind of align you and orient everybody. And what you're seeing here is these big dots are the mean location where we find the neuron across these 10 animals. And the ellipsoids that you see around them, that's the positional variability that we see, right? So that's where we might find a neuron, anywhere from here to here, so to speak, within this particular circle, right? And so what I want to point out here is that while some of these neurons have extreme stereotypy in where they're located, for example, this purple neuron here, AVA, is always found in this very tight location. Other neurons have really a large amount of positional variability, as you can see here in the retrovesicular ganglion of the worm. That's what all these neurons are. And they might really kind of shuffle around, and thus their position alone is not enough to give away their identity. And why that's the case, why we actually see all this positional variability in some places but not others, remains unclear. We try to figure it out, but we were unable to determine a particular cause for that. So how do we actually build in a cell ID strain? There's only one simple rule for color barcoding a bunch of cells so that you can distinguish them. And that is that neighboring cells, or neighboring neurons in this case, must be distinguishable. So I'm going to explain what that means. Oh, is there a question? No, okay.
Not yet, no.
Okay, great. I'm just going to take a sip of water also. So if we have these three cell types, A, B, and C, if we have no coloring, we are unable to see them, right? So the very first thing is to bring in fluorescent colors into them. Now, if two cell types have the same coloring, for in this example, A and B are colored in green, we can't tell them apart, right? So, if you see some green neurons and you ask me, hey, I'm looking at some green neurons, what are they? Well, I'll tell you, look, A and B are colored the same. They're both green neurons. I don't know if you're looking at cell type A or cell type B. But once we bring in different colors, now we know exactly what cell types we're dealing with, right? So if you come to me and you say, hey, I'm looking at a green cell type, what is that? Well, there's only one green cell type in this particular location. It's cell type A, right? So we have no ambiguity in knowing what cell type we're looking at. And if we have different regions, for example, maybe the head of a worm or the tail of the worm, or two ganglia that are separated, from each other, we can reuse the colors. And why? Well, if you come to me and you say, I'm looking at a green cell type, what is it? I'll just say to you, are you looking at the head or the tail of the worm? And you say, oh, I'm looking at the head of the worm. What's that green cell? I can tell you definitively that's cell type A. There's only one green cell type in the head in this case, and it's cell type A. And that's basically the process. So now we have to color all these neurons in such a way that they're distinguishable from their neighbors. Okay. Now, before I move forward, I'm just going to get into a very confusing concept, which is reimagining the actual colors and telling you that red is the new green. So I'll explain that. Every time that you've seen GFP in a paper on your screen, unless you're looking directly through the scope with your own eyes, that green is a pseudo color. And what I mean by that is ultimately you have some illumination that comes through from your sample. You spin a filter in front of it that gets the wavelength of color that you're interested in, and only lets that through. It doesn't let in any other wavelengths. And your camera just measures the intensity of that light coming through that filter, right? And then it calls it a channel. It calls it channel 1, and then there's channel 2. It doesn't know green. It doesn't know red. It doesn't know anything. Later on in your software, you tell it, hey, the GFP channel, which channel 1, let's say, is green colored, and you assign green color to it. But you've probably seen in various journals, sometimes GFP is colored magenta, just because it's easier to distinguish that, in the backdrop against some other colors, right? And so ultimately, you can pseudo color any color that your camera sees any way you want to, and thus color GFP anything other than green, right? And the key thing here is because I need to build a palette of colors, and I need red, green, and blue as barcoded colors, GFP, which is part of the mix here, is not going to be color green in this presentation. It's always going to be white. And that'll all make sense in a second. So when you see green, you're not seeing GFP, you're seeing a totally other fluorophore, which happens to be red, and I just switched to color green in software. Okay. So first and foremost, the more colors, the merrier, right? With one fluorophore and a stable reporter that drives that fluorophore in a particular set of cells or a particular set of neurons, I found that in the worm, we get about 3 distinguishable IDs. In some cells, it's weak, and some cells it's going to be maybe medium colored, and other ones it's going to be bright. If we get that range, sometimes we just see bright, cells, sometimes only weak. But we've seen basically that we get we can distinguish by eye weakly colored cells, medium colored cells, and bright cells. And from that perspective, if I've just got that in place, and you come to me and you say, hey, I'm looking at a green colored cell, what is it? I'll say, is it the brightest one in the mix, or is it the weakest, green color that you're seeing amongst all the cell types? And if you come back to me and you say, it's neither the brightest nor the weakest, somewhere in between, I would call it medium, I can tell you that's cell type B, right? And so on and so forth. And once we start to bring in other colors, now we have a combinatorial code. What I mean by that is any particular neuron, in this case, or cell, can express weak green, medium green, bright green, or no green. So we got 4 different levels of green that we express, times 4 levels of blue once we bring blue in, right? Weak colored blue, medium colored blue, bright blue, no blue, times 4 levels of red. And so we have 4 times 4 times 4, which we can express in neuron. It could be only green, or it can be weak green, bright blue, weak red, or however it be, right? And through that, we get about 60 different, well, it's closer to 63 different IDs, barcodes that we can apply to ID different cell types. Now, that's all theory, and I'm going to tell you about practice. The way I put colors in particular neurons is I use reporters that target subsets of neurons. And so they put green in some subset of neurons and blue in another subset of neurons that overlap. And now we've got green and blue in some neurons, neurons that are only green, neurons that are only blue. But ultimately, I cannot target all the neurons as subsets. And so when I was done, what I had is a bunch of neurons that had no color because I just didn't have reporters that reached them. And so for that reason, I needed a fourth color and a pan-neuronal reporter. So we have reporters that drive fluorophores in all neurons or drive anything we want basically in all neurons. In this case, I'm driving a fluorescent protein in all neurons. And I use that with the fourth color so that every single neuron is labeled with this fourth color, which I'm showing as white here. And then in addition to that, some neurons will also have a little bit of green, a little bit of blue, a little bit of red, or some amount of, these basically so that they can be distinguishable from their neighbors. And now I've told you about four colors, and that's still not enough. Because ultimately the reason we're using this is for what I told you before, which is to look at expression of particular genes in different neurons or to look at their activity, right? So we need a fifth color, which is going to be the signal. And the fact is that everybody in biology, for the most part, uses GFP at GCamp, which is a version of GFP. They both have very similar spectra. And in fact, GCamp was created from GFP. They've already locked that into place. So I can't use that for my barcodes. I do have to respect the fact that people already have a lot of strains with it. And so I have to make room for it. So ultimately, this is a 5-color method with 3 colors that are going to give me the red, green, and blue color, a barcode. A fourth color, which is going to light up all neurons, and then a fifth one, which is a signal, which is going to be GFP or GCamp, which we cross into the NeuroPAL strain. So it doesn't contain this, it can be crossed to it, and you can see the signal alone. And again, white from now on is not going to be, it's going to basically be the GFP and the GCamp or the pan-neuronal reporter. I'll let you know which one it is, but green is not GFP, so don't get confused. Green in this presentation is never GFP. It's this red color called CyOP, which you don't need to know about. So the challenge was to find 5 distinguishable fluorophores. And that's what I did. And these are they. So they're this MTAG-BFP2, GFP or GCamp, CyOP, TAG-RP, and M-Neptune. This is how we excite them with these 4 lasers, although we can actually excite them with just 3 of them. And then they emit here. And the key thing here is that I don't want to get into too much details, but there's this factor of basically finding fluorophores that work with filter sets that exist and are being sold. It's not that easy. And ultimately, the solutions to the problem are really constrained. But I found 5 fluorophores, 4 fluorophores that work with GFP and GCamp, for which we have filters that can distinguish them, and they're available from various companies. And so every microscope works with this technique, and you just order these filters. Filters are relatively cheap. You can use this on your compound scope, your dissecting scope, confocal, light sheet. It doesn't matter. All you have to do is buy these filters and spin them in front of a camera or a photomultiplier tube and basically get what you want out. Right? Photomultiplier tubes tend to work differently. So, okay, now let's get to how we actually take that, which I just showed you, and basically use it to give us these neuron-specific or cell-specific barcodes. How do I combine these reporters to get the barcodes that we want? And how do I paint with neuron-specific reporters? Right? So I told you before that if I have just one color, in this case red, and I have it weak, in some neurons in brighter in other neurons, I can distinguish those because of the level, the intensity of that color. So here what I'm showing you is reporters in rows. These are the different reporters that target color to different neuron subsets. And you'll note that there's a pan-neuronal reporter targeting color to every single neuron. That's the 4th color. And then here in columns, I have the different neuron types that I want to target with color, right? So in this case, if I just had this and you said, what's this weak red I'm looking at here? What's this weak red cell? I can tell you, well, that's neuron number 2, right? Just as I told you before. Once we start to bring in more colors, we get this richer palette to work with, right? And so in this case, we'll have basically reporter 1 targeting blue to these different cell types and reporter 2 targeting green to them. And what I just want to focus you on here is 2 neurons, which I think are interesting. Neuron 5, reporter number 2 is driving green in it, and that's the only reporter driving any color in it. And that neuron's then going to look green to your eye because it's just expressing green. Neuron 8, on the other hand, is targeted by reporter 2 and thus has green in it, but it also has reporter 3 driving red in it. And that combination of green and red to your eye is going to look yellow. And that's how we get kind of the simpler color code. And once we bring in the different intensity levels, then we really get a rich rainbow of colors. Pretty much that RGB color palette that you see on screen, we can recreate effectively within the neurons themselves. Okay, so now the magic part, which is still a miracle to me that it worked. I went through 130 reporters that target different subsets of neurons. I chose 41 that are stably expressed. So we see them at all stages of development the same way. And so I biased myself towards a stable color code, a stable set of reporters that don't change throughout the life of the worm. I injected them into the gonad of the worm, or actually I had a technician inject them to the gonad of the worm, and she's fantastic. Someone who works in Oliver's lab named Chi, and she's wonderful. Without this work, without her, this work couldn't have been done. And through some miracle of the worm, those 41 reporters get combined into a single transgene, and they're all there, right? And what I want to just tell you is, well, I'll get to the punchline in a second. This is what they look like. So here are the 41 different reporters that I used in rows here in the pan-neuronal reporter on top. And these are the different neuron types in the worm listed by ganglia. And here, these thick lines separate different ganglia. What I want you to notice is this bottom row here. This bottom row is the real colors that we see in actual NeuroPAL worms. I just took the eyedropper tool with Excel and pulled these colors off of the actual images. So this is what they look like. And you'll note that in each ganglia, the different neurons have different colors, and we can tell them apart because of those colors. And the reason they have those colors is because of the reporters that basically label certain neurons with color. So the reason this neuron here, which is called SETV, doesn't really matter what its name is. It's a ventral cephalic neuron, but again, that doesn't matter. The reason it's purple is because a dopamine transporter reporter is driving red in it, and a cholinergic receptor reporter is driving blue in it. And the combination of blue and red to your eye looks purple. And we'll return to this concept in a little bit, but the key thing here is that the identity of what you're looking at in terms of the color is the live visualization of the identity of the neuron, because we have terminal identity reporters driving that particular color. And if the color gets altered for whatever reason, because the environment changed, we have a mutation, we'll know it because we expect to see certain colors, certain neurons of particular color in particular locations, and we no longer see them. So we know something went wrong. But all that we'll return to in a second. This spreadsheet here, this is the barcode, the live barcode that gives us the different neuron types. And it turns out that this technique is robust. I don't know why, part of science always is luck. But the key thing here is that every time I injected this cocktail, I got worms that had exactly this coloring, as you see here. And ultimately, what I did is these transgenes are extrachromosomal, right? So they just float around and they're lost eventually as a result. But we gamma radiate the worms. We break apart their DNA, and sometimes the worm's machinery, basically, it's cellular machinery for DNA repair, basically is like, oh no, what happened? My DNA is fractured. And in reassembling it, they take up the transgene and then put it in. They're like, oh, this belongs inside me, I guess. I'll just put it into my genome. And now it's integrated. And now it's expressed in all worms, and it's not lost because it's in one of the worm's 6 chromosomes. And all those worms look alike. So I inserted this transgene into multiple locations on different chromosomes, and all those worms looked alike. Okay, so this is what the NeuroPAL looks like. This is a NeuroPAL head here. These are the different neurons in it. And this is just to show you that the coloring works across the entire body, nervous system of the worm, right? So in any particular location, the ganglia which are encircled, basically we can tell the different neuron types through those coloring. And sometimes you see 2 colors that look very much alike here in that ganglia. Well, these are neurons of the same type. This is a DB neuron here. No, sorry, this is a lateral ganglion. So this is, in fact, a neuron in a different ganglion here that's kind of sneaking through. But generally speaking here, the neurons are distinguishable from their neighbors. So in this example, there's 2 blue neurons, and these are 2 VD type neurons. that really doesn't matter. I'm getting into too much detail. Okay, now that you understand the method, I want to tell you about 3 NeuroPAL cell ID applications and the exciting results that came from them. The first one is identifying gene expression. So I told you before that we can identify gene expression patterns using the NeuroPAL. I gave you an example in which we had a GABA receptor expressed on the surface of a variety of neurons. We crossed that to the NeuroPAL, and thus we're able to know which neurons express that particular GABA receptor, that gene. And this is, by the way, this is GFP in white here. The NeuroPAL doesn't contain GFP, and when we cross the NeuroPAL to it, you can see where that GFP is located. And so what I did with that, first and foremost, is I completed the metabotropic and Gaba connectomes. I'll make sense of that for people who it's probably confusing actually to many people, even those potentially who are in the field. So just a quick review of the nervous system. In all animals, basically there's just 2 flavors of neurotransmitter receptors. There are these ionotropic receptors, which mediate fast direct communication. So they open up, ions flow through them, and that basically activates or inhibits the neuron. And then we have this other type, which is called the metabotropic receptors, and they're slower. They have these second messenger communication effects that lead to a variety of additional downstream effects, but they have long-lasting effects, and they can even basically change a little bit the state of the nervous system. And for that reason, I wanted to focus on them, and also because they have over 80% homology to their human orthologs, so they're very similar. They're conserved basically, and they're similar to their human counterparts. So what I did is I identified the expression of all metabotropic neurotransmitter receptors using NeuroPAL. And then what I did is I used that to fill in the connectome. So I told you about this earlier, but basically since the 80s, we've known about the synaptic connectivity of every single neuron in the worm's nervous system. And that's what's on display here. So these circles are neurons, and then the edges between them, those lines basically are the connections that we know about, the synaptic connections. Some of these are GAB junctions, and some of these are chemical synapses. In this case, I think I'm showing both. And through really nice work from a postdoc in Oliver's lab, where I did my own postdoc, we know which neurons here in this small network basically release GABA. So these neurons in blue here, these are the GABA releasing neurons in this network. You'll note that only a small part of the network releases GABA, about 10% of this network. And these are the neurons that express GABA receptors, both ionotropic and metabotropic GABA receptors. So I filled in where the metabotropic GABA receptors are, and some nice work before me had filled in where all the ionotropic GABA receptors were. And as a result, we now knew where all the GABA receptors were and all the neurons that release GABA, and we were able to complete this GABA connectome, so to speak. So all the neurons that release it and all those neurons that are synaptically connected to those neurons that release GABA and receive it and listen for it. And the surprising finding, number one, is that there are a lot of neurons that express GABA receptors on their surface, but even more so surprising here in red are these neurons that are not synaptically connected to any GABA releasing neuron that express GABA receptors. So these neurons are listening for GABA, or they have the potential to listen for GABA and receive GABA signals, but they're not synaptically connected to any neuron that releases it. So if in fact they are receiving GABA and responding to it, what that GABA must be extrasynaptic. In fact, they're listening in, so to speak, on their neighbors' communications as a result. And it looks like there's a striking amount of the nervous system that is basically listening for this extrasynaptic, or it has the potential to receive extrasynaptic GABA. And mammals, I think it's often called volume transmission, but it looks like there's a large portion of the nervous system that basically is able to receive those signals. And so you might wonder, for those people in the field, well, how much of the nervous system is able to receive these signals outside of the synapses? It turns out that irrespective of whether or not we're talking about ionotropic or metabotropic GABA receptors, 30% of the neurons express them and are not synaptically connected to any neuron that releases GABA. The same is not true for the cholinergic and glutamatergic metabotropic networks. There we see far less spill, over or extrasynaptic communication, potential for extrasynaptic communication. And then I think the next natural question for people who are interested in neural communication is, well, what are all these neurons that are listening for GABA that aren't synaptically connected to a neuron that releases it? and it looks like overwhelmingly they're sensory neurons. And so one hypothesis might be that the nervous system is basically has this as the means of kind of gating stimuli that come in. So if you have a bunch of stimuli coming in now to the nervous system, you have maybe a lot of GABA spillover that's silencing and creating a threshold basically for other neurons in the system. And any new stimulus that's coming in order for it to be big enough for in order for it to create a response in the worm, it has to be a large enough stimulus to overcome the GABA inhibition that's already taking place in this nervous system. I'm sorry if I've confused people who are not in this field. I can answer questions about that if people have. The second thing we did is ID the expression of all 102 worm homeobox genes. So these are a famous set of genes that are conserved across all animals, and they were discovered because they lead to extra segments in a variety of animals. So there's some repetition that takes place as a result of mutations in these genes. We believe, or our hypothesis basically is that they essentially give the different neurons their identities, right? So we have these pan-neuronal reporters, all neurons express them, but ultimately different neurons have different neuron types. They have different roles. Some are sensory, some are interneurons. They're basically doing different types of computation. And we believe that the homeobox genes, these homeodomain proteins, basically are largely responsible for giving those different neurons their different types, for putting their specific inventory into specific neuron types. And so here are the homeodomain proteins in rows, and here are the different neuron types in columns. And what you'll note is that while some of these homeodomain proteins are expressed in every single neuron, others are expressed in just a subset. And this work, by the way, was led by Molly, a PhD student in Oliver's lab. She did fantastic work, and I was really happy to be a part of this. This is her here. And what she did is ID'd all 102 homeobox genes using NeuroPAL. So the work was only possible because we had NeuroPAL. And she found that 24 conserved homeobox genes, so 24 homeobox genes that are present in worm and in human beings, uniquely identify all 118 neuron classes. And so again, the hypothesis is that in our case, we believe, I should say this first. In other animals where people have looked, so not our work, so in flies and in mice, we found similar things. The scientific communities found similar situation in which basically the homeobox genes are the family of transcription factors that best discriminates different neuron types. And so our theory here basically is that homeobox genes acted early on in evolution to give us a diversity of neuron types that we see in all animals. And I don't know if I should get into this. I'll get into it. A lot of people might say, okay, well, NeuroPAL used it to identify all these homeobox genes. Why not just use single-cell transcriptomics? In fact, in the worm, we've done single-cell transcriptomics for all the neuron types now. So we know their transcriptional inventory. But still, the NeuroPAL is actually quite effective in giving us the ground truth, first and foremost, to call thresholds for what is, noise in that transcriptome that we have and what is real. And also, there's a variety of things that take place in the pathway from the point at which we have transcription to the level at which we actually have protein. We have post-transcriptional modification, and then post-translational modifications that take place as well. And in fact, the NeuroPAL is still very good at letting us know ultimately what genes lead to protein in the final, in the cell. And we don't always see correspondence between what we see in the transcriptome and what we see as actual protein expression. Okay. So the second NeuroPAL application I'll tell you about is identifying cell fate. And here I'm just going to go back to the spreadsheet I showed you earlier. The reason the neurons have the color that they have is because of the set of reporters that are driving colors within them. And these reporters are for identity markers for the neurons, right? So in the case of CEP-V, which I told you about before, this particular neuron, name doesn't matter, is purple because this neuron has within it a dopamine transporter. So it's a dopaminergic neuron. And it also has a cholinergic receptor. And that cholinergic receptor here has been co-opted also to drive blue coloring in the neuron, and the dopamine transporter is driving red in it, right? And if we bring in a transcription factor mutant, and we, for example, see that this neuron loses one of its colors, right? It loses blue or red. We know exactly what gene it no longer expresses, right? And we know the identity or the fate change that's taken place, right? What has changed about this neuron? And we can do that for all these different neurons when we lose coloring. And if we gain coloring, we can even hypothesize about what gene might now be expressed in that neuron. And so we can use the coloring as a live visualization of cell fate to see what effect particular transcription factors or other environmental manipulations basically have on the cell fate of particular neurons. So I'm going to take you through that. That's probably a mouthful, probably a little bit confusing, but it all makes sense in a second. This is a conserved transcription factor called ER1-PLZF. It's in human beings. Name doesn't matter. But really what I want you to focus on is here it is in white, GFP. And this is the head of a worm. It looks like it's in every single cell in this worm's head. And this is it in the embryo. And again, it looks like it's in every single cell in the embryos that you're seeing here on the right? So if you wanted to kind of determine where this particular transcription factor acts or which cell fates it's responsible in, well, it would take you a very long while to figure that out because it could be acting everywhere, or maybe it's acting just a subset of these cells that you're seeing here, maybe in none of them, right? But you'd have to test all these different hypotheses. The NeuroPAL gives us an unbiased way to screen across, or relatively unbiased way to screen across the entire nervous system for cell fate transformations, or where particular transcription factors act. Now, I'm going to take you an example with ER1-PLZF. This is a wild type NeuroPAL. You can see again all these different neurons in the head of the worm. And I just want you to focus on these two neurons here. Their name doesn't matter, but they're the dorsal and ventral RMEs, which are blue. They are blue because there are 3 reporters that are conserved. So these, for genes that are conserved. So these genes are in other animals as well. And these reporters for these genes are driving blue coloring in these neurons, and thus they're blue. And also they express a paneuronal reporter in white, right? So they're neuronal, and thus they express in white this neuronal reporter. Once we cross in the OR1 mutant, which I showed you previously is expressed everywhere, you would say, oh, isn't it going to mess up the entire worm, right? So we have a mutant that doesn't have this particular ER1 transcription factor in it. Surprisingly, it looks very much like the wild type, right? In fact, you might guess, well, it looks like it didn't have any effect at all. The striking thing here is, in fact, of all the places it could have had an effect across the entire nervous system, it seems to act just in these 2 neurons. There's a little bit of effect in a few other neurons, but it's neither here nor there. But here, the effect is quite striking. The blue neurons that you see here in the wild type are no longer present, right? And so we immediately know that this transcription factor basically acts in putting those particular genes in these neurons. What are those genes? It turns out that one of them is a Gaba synthesis enzyme, GAD, which is UNC25. And in fact, these neurons are no longer able to synthesize their own GABA as a result. Now, you might say, okay, well, how do you know that these neurons didn't just die at birth, or maybe they're not there anymore, maybe they moved? And it turns out, well, that's where the paneuronal reporter kicks in. We can see still that they express a paneuronal reporter, so these neurons are still of neuronal fate. They've just lost particular aspects of their cell identity, their fate here that we can basically pick apart. And this is just to show you the profound loss of blue, basically quantitated. These neurons lose, most of the animals have no blue in those neurons. And this is just confirming it with another reporter, right? So if we bring in something totally different that has nothing to do with NeuroPAL, we find again, we can confirm that we lost this particular gene UNC25, as well as the other genes that the NeuroPAL shows us that we've lost in these particular neurons. Okay, the other exciting thing I want to tell you about is identifying cell fate, particularly identifying neuronal duplications and looking at this really exciting thing that we found called just-in-time differentiation. It all makes sense in this next slide. This is work, by the way, that was led by a colleague of mine, Tessa, who was also a PhD student in Oliver's lab, and she's also fantastic. And I got a chance to be a part of this really nice work that she led here. And so the first thing I want to show you is this. This is a wild type NeuroPAL worm. And I told you before that the neurons are distinguishable from their neighbors because they have a different color than their neighbors. Here we've crossed it into a particular mutant that leads to duplications or reiterations in the lineage. So every time the worm goes through a molt, a different life stage, it basically, unlike the wild type, in this mutant, it reiterates a division, a cell division. And in the end, it's left with 3 neurons that are PVW when it was supposed to just end up with 1. And we can see that here. We see 3 cells, 3 neurons that look identical. next to each other. And so our immediate conclusion is, hey, maybe a duplication took place. And this is what duplications look like: 3 PLNs and 3 PHCs in this mutant worm. The other thing is this. The neurons take on their identity markers, their colors, when they differentiate, right? So when they've reached their mature state, their fully differentiated state, then they have the coloring. And what we found here is that, well, we knew about a variety of neurons through work that was done in, I think, the 80s as well, pretty sure, yeah. That a variety of neurons that are present in the male are born at different stages of development. Some of them are even born in the embryo. What you're seeing here is a male tail from an early fourth larval stage worm. So it's a male, but it's not yet sexually mature. You'll notice this black spot here basically has no colors within it. And there are a variety of cells here that are going to be neurons very soon. They were born at different stages of development, but they're kept in stasis, awaiting sexual maturation. In a few hours before this male basically becomes a sexually mature male, we see all these colors turn on. So we see this just-in-time differentiation. These cells become neurons just in time for them to be needed for males basically to have sex. And that's ultimately their role, is essentially allowing the males to copulate. And so we see that the male basically turns on its neurons just in time for their need in sexual maturity. And we see that using the NeuroPAL. And lastly, the third application I want to tell you about is identifying whole brain neuronal activity. And this is the work that I'm super excited about and the whole reason I built it. This is a NeuroPAL worm in a microfluidic device. The flashing lights that you see, or first of all, you see the colors and we can identify the various neurons. The flashing lights you see here, this is GCAM. It's showing us the activity of the various neurons. It's basically a GFP-based sensor for calcium that shows us neuronal activity. In the top right corner here, you can see the stimuli that we're applying, and I'll get into those details. But anytime a stimuli comes in, you can see a variety of neurons basically responding to it. And I've labeled a large subset of those neurons, but not all of them because I didn't want to crowd out the image. But ultimately, you're seeing basically a worm responding to a variety of stimuli, and you're seeing what's taking place in its head. This is a collaboration with the Venkateshalam lab that's at Northeastern now, and in the Samuel lab, which is at Harvard. And I've sped it all up. And here, this is just one head. Just to show you how much data goes into this, it's about 190 neurons in this head of this worm. I've ID'd all of them, and that's what you're seeing here, the neuronal activity. So the different rows are neurons. The activity that you're seeing, the columns are basically the activity over time. I'm just going to zoom into this neuron I told you about earlier, AWC left, and its responses to salt and to odors. It's an olfactory neuron, so it senses odors, but it also seems to be responsive to salt here. You can see that it's basically loses calcium. Sorry, it responds to these things through what we believe is inhibition as a result of what we see in these traces. But I have 190 neurons here in this one head. And in fact, the work I'm going to show you now here, which is possible only because of the NeuroPAL, uses 21 heads and also 21 tails. So multiply this times 21 and you'll get a sense of how much data is going into this. We built a special scope for imaging these worms' heads and tails. Basically, we have an objective. The worm is stuck here in a microfluidic device. We have stimuli and buffer kind of flowing by its nose, and we can give these different stimuli. We put the objective either over the head of the worm, and thus can tell what the neurons are doing in the head of the worm, or over its tail. And then we turn on these 4 colors that you're seeing, sorry, these 4 lasers here to get the different colors, but it turns out we only need 3 of these lasers. We've kind of figured out a nice way to get by without the 4th laser. So we turn them on, get the NeuroPAL colors out, learn the identities of the neurons, and then we turn them all off except for the 48 laser, which we use to record the activity of the GCAM. And the stimuli we're using here are a large amount of salt, which is repulsive to the worm. It's as if I would have dumped a huge amount of salt on your tongue. The worm hates it and it runs away from it. And then 2 odors that the worm likes, because it's as if it's smelling food in its location. It's attracted to these odors. One of them smells to us like butterscotch and the other one like caramel. So a lot of us are attracted to them as well. And then we look to see what takes place. Now, this is some earlier work from a paper I was on in 2015. Here, the worm, this is just one worm in a microfluidic device, and we're observing what the neurons are. It's immobilized, but we can tell you what the fictive behaviors are based off of which neurons are active, right? So we can see it thinking, if I can anthropomorphize the worm, that it's going forward, reversing, turning one way or another, right? And here what we found, in fact, is that the neurons, all these neurons in the head of the worm have a very low dimensional representation. And in fact, 65% of, three principal components, or three low dimensional components, basically account for 65% of the variability in between different neurons. And I think based off of that, a lot of people concluded at that time, well, if you only need, if 65% of the variance in neuronal activity isn't basically accounted for by just three principal components, why do you need to look at all these neurons in a worm's head? Can't you just tell what's going on there just off of a few of them? Because there seems to be a lot of overlap in terms of, or correlation in terms of what they're doing. And what we found here in this new work is that once we bring in these different stimuli, we see a very high dimensional code or response. And in this case, now to account for just 65% of the variance in neuronal activity, we need 40 principal components. And in fact, here what I'm showing you is that it's a slow and steady climb to account for all the variance in different neuron activity that really requires nearly all of the neurons' activity to understand what the response is that's taking place here, right? So we really need to see the different neurons and know their types. The other thing here is that in this previous work, what we're showing you in the low dimensional space is just one worm. And why? Because the dimensions are the different neuron types. We don't know what they are. We didn't know what they were in this previous work. And so we could only project one worm into a low dimensional space at a time. But here I'm projecting 21 worm heads into one space and then collapsing them into a lower dimensionality. And what I want to show you here is that this thick line in the middle is what those different worms have in common in terms of their response to different stimuli. And the tubing around them is the variance we see between different animals. So you can think of it as the individuality that we see in different animals. They don't all respond the same to these stimuli. I just want to stop, by the way, and ask if there are any questions, or and hope also that the feed is still active, because Zoom can sometimes drop out. Okay, perfect. Yeah, excellent. So now I'm going to show you one of the most striking things. I think a lot of people know about the worm connectome, and I showed it earlier. We knew this map of synaptic connectivity since the 80s. We knew about the electrical connectivity, so to speak, the gap junctions, and the chemical synapses as well, right? And so you might say, okay, well, you've had the circuit diagram, the connectome, since the 80s. Why don't you know what all the circuits in the worm do? And in fact, why do you maybe even need to see the functional activity, right? Why do you need to see the dynamic qualities of activity taking place in these neurons? Can't you predict it from the circuit itself? And that was a question that I was quite interested as well in. And so here, what I'm showing you is the correlation between the dynamic functional activity that we see and the synapses that we know about, right? The weighing of those synapses. And it turns out that they are very poorly correlated. They're in fact almost not correlated at all. In the case of the electrical synapses, that's the best we can do. About 3% correlation between the synapses we know about, the connectome we know about, and the functional activity. It's even worse for the chemical synapses, irrespective of whether we're talking about the head or the tail. It's just terrible. And that really motivates the need for looking at the dynamic quality of the neurons. And I think some people will not be surprised. They might view this as, hey, look, the map of synapses, sure, that's the hardware, but there's software running on top of that. There's neuromodulation. There's no reason the two have to look alike. But we actually think there's more at play here. First, I showed you earlier in the talk the potential for extrasynaptic Gaba signaling. So there's potentially a lot of signaling. There's signaling there that's taking place outside of the synapses. There's this concept also of the dark connectome, the connectivity that we didn't see with when we were just looking at synapses. And that's been largely pioneered by my PhD advisor, Bill Schaefer, in which he has found, essentially in his lab, and this is some work that I got to be a little bit of a part of as well, that basically when we start looking at the monoamines like dopamine, serotonin, and things like that, and neuropeptides, which people might be familiar with, oxytocin and other neuropeptides, the nervous system, at least of the worm, is largely extrasynaptic communication, right? It's this dark connectome that we don't have access to. A lot of neurons are communicating wirelessly here. We weren't able to see it in electron micrographs. And the only way we might have access to it, at least for now, is looking at the functional activity. Maybe eventually we'll piece together which neurons communicate wirelessly like this, but we don't have that in place. And that might account for all this, the lack of correlation between the two views of this connectivity. Very quickly, just to wrap up, we built software. This is really due to a collaboration with Liam Paninsky's group at the Zuckerman Institute. They're fantastic. And I should also say that Vivek Tashalam and his lab were fantastic. And Ervi Samuel was fantastic. I mean, this was a project where I got to work with some of the most brilliant people I know, and it was so much fun. But the Paninsky lab here, we built together with them basically the software that automatically detects and IDs neurons in the worm, right? So you might say, okay, well, 190 neurons, that's a lot of neurons to go through, I mean, and then 21 heads, that's really a lot. And then add the tails, that's a lot of work. So we built this software here, and you just input images of the head of the tail of the worm, and it detects the neurons, and then it identifies them and gives some probability for its different IDs. Here it's identifying the RIM neuron with 94% confidence, and it's in fact right that this is indeed the RIM neuron. And if you're wondering about its accuracy, well, by design, this whole system of NeuroPAL, humans can have 100% accuracy on, right? That was by design, but the software does really well on it too. In the head, its auto ID accuracy is 86%. In the tail, it's 94%. And in fact, if you manually label about 10 neurons, it uses those predictions to improve it uses your annotations to improve its predictions. And with 10 annotations in the head, its accuracy rises to above 90% for neuron classification. The other thing we did is we built software basically to bring NeuroPAL to any tissue in any organism at all that allows transgenesis. And so here we built this. This is really nice work from Erdem Varol, who now has his own lab that he's starting up at NYU. Really fantastic guy, but he was part of the Paninsky lab at the time. And the idea is you say, okay, I am working not with a worm, but with fly, for example. I have just here's a list of reporters I have that can target different cell types. And I don't have the luxury of doing 41 reporters with 3 colors. I can do 3 reporters tops in the system. Give me the best design you can to distinguish different cell types with color. And this program will put out approximately optimal solutions. So these recipes of how to combine different reporters with different colors to achieve distinguishable cell types with coloring. And it'll spit out a bunch of recipes for that, so you could try them out and see which one works best for you. And here it is, just simulating a NeuroPAL and doing really well and with 41 reporters and the top left. But in the bottom right, we said, hey, you know what? 41 reporters is too much. No one's going to work with that. There's just too much work. Can you do it with 3 reporters? And in fact, with 3 reporters, it still does really well in terms of distinguishing different neuron types with color. And so in summary, I showed you how I combined these color reporters to produce the Neuronal ID strain, use it to ID gene expression for the connectome, for the metabotropic connectome and the Gaba connectome, with surprising results for potential Gaba communication, and also for the homeobox genes to identify where they are at. That was Molly's work. Identify sulfate changes, in this case, looking at in an unbiased way across the nervous system to see where a particular transcription factor acts, and also this nice work from Tessa in looking at just-in-time differentiation and cell duplications. And then lastly, identify neuronal activity as a collaboration with the Venkateshalam and Samuel labs, basically finding that in fact we need a lot of the neurons basically to understand the responses of particular animals to different stimuli, and also seeing this lack of correlation between the circuit diagram we know about, the synaptic circuits, I should say, and the actual activity that we see. And then software to automate an ID and build the ID strains for Liam Paninsky's group. And then really quickly, future directions. I want to bring this technique to flies. The idea would be a little bit different here. Not going to target every single neuron in the fly, at least not at this early stage. There's just too many of them. But the idea is to create 2 lines, one that puts coloring into particular cells and another one that drives that coloring using this binary expression system for those who are no flies, in which we use GAL4 and the GAL4 upstream activating sequence, and LexA and the LexA op, and the Q system as well to drive the different colors. And what you do is you have one line, which is a universal coloring line. It doesn't put color anywhere, but when you cross it to another line, this is the idea, design behind it that I'd like to implement. That line contains the enhancers that will drive coloring where you need it by basically using the binary expression system to activate the colors in the universal reporter line. And so the idea I had was basically to take some old work from Michael Adams' group, in which they labeled different ecdysis-specific neurons, these neurons that are involved in the molt to adulthood. They labeled them with GCAMP and looked at them individually, but in this case, combined them all in one animal with different coloring so we can tell which neuron is which and look at them simultaneously. And then if you're interested in more details, I have a lab website. We're always recruiting good people. If you're interested in joining us, I would love to have more people in the effort. And what I'm specifically interested in my lab is not just bringing NeuroPAL to other organisms like fly and mice, but also looking at development, looking to see how neural activity changes throughout development from these young animals to sexually mature animals to create age-appropriate behaviors, right? So why do males behave the way that they do and females behave the way that they do? Why do young worms behave very differently than older worms? How's that done at the level of whole nervous system, neural communication? And just to acknowledge all the people involved in this work, members of the Hobart Lab, as you saw, played a large role in this and also in helping me choose the reporters that you saw, giving me great feedback. The Paninsky Lab worked on the software that you saw, and the Samuel and Venkachachalam labs worked on these are all collaborations, by the way, worked on the whole brain imaging scope and the analysis of it with me. And I guess now I should take the questions.
Absolutely.
And thank you for staying with me for this long.
This was a really cool talk. This is that's super cool. So I'm just going to jump into the questions. You've got a good number of them here. So the first one here is basically asking, there's only 302 neurons in the worm. Did the animals not have spontaneous neural growth and pruning?
That is a fantastic question. So the answer is I've oversimplified it. The worms are born with 222 neurons. Mm-hmm. So those are the embryonically born neurons. And then they add more neurons. I should go all the way to the beginning of this thing. They add more neurons at their different life stages. So they have these post-embryonic neurons that they add into the nervous system. We don't know how that's done. I mean, it's pretty amazing, right? Mm-hmm. Much like us, their brain, so to speak, or nervous system is growing larger as they add in these new neurons. And at the stage in which they transition to adulthood, the females will add on 8 neurons for egg laying, but the males will add on another about 30% more neurons, basically, for copulation, for sexual behaviors. So, yeah, and there is this, while the neurons are stereotyped, meaning that at these different life stages, these worms will all have the exact same set of neurons, with these names, that we have for them, essentially, because their morphology is the same from one worm to the next. In males, there's these mysterious neurons called the DXs and EFs, and it looks like there's some variability. A worm can have 2 DXs or it can have up to 4 DXs. It can have 2 EFs or up to 4 EFs, male worms, and that's the only time in which we actually see any stochasticity in terms of the number of neurons that a worm has.
Okay. Let's see. What characteristics determine a neuron type?
Yeah, that's a great question. This gets into this gets us into territory that causes actual fights between people.
So the best kind of territory then. Okay.
I love it. I don't shy away from these fights. I told you that the worm has 302 neurons. I'm telling you right now that I lied to you because I believe the worm only has 300 neurons. Ooh. Some people won't admit to it.
Hot take.
But these two neurons here are that people, where the debate really lies is these neurons called CAN. And I talked to John White, who did the original work. I said, why did you call these neurons? They don't have certain characteristics of neurons. I'll talk about those in a second. And he said, at the time when we sectioned the worm, they were running up along another set of neurons in terms of their projection, or the wiring was running up against the wiring of what was definitively another neuron. And it was just a really long wire, and they didn't have enough sections. So they just said, maybe it is, maybe it isn't. And they just called it that way. So what makes them not a neuron, or in my opinion, at least, is as follows. They don't release neurotransmitters onto any other neuron, basically. So they have, I think they have gap junctions, but they don't release, right? They don't have synapses, presynaptic modules from them onto anything else. When you kill them, the worm dies. That's something that's more of a feature of a gland cell. And aside from that, their ultrastructure in the EM doesn't look neuronal-like. And moreover, lastly, they don't express certain genes that all neurons, or it's a better way to put it. I told you about the pan-neuronal reporter. The pan-neuronal reporters we have drive neuronal, and then we have many of them. They drive expression in all neurons except for these. If you want to hit these neurons with the reporter, you also have to hit things that are definitively not neurons, like muscle, hypodermis, and glands, and the like. So, from a perspective of genetics, ultrastructure, function, synapses, they are not neuron-like. Now, I said all that, and I said that to Oliver, who was my postdoc mentor. I said, these aren't neurons. I said, by the definition of what I think is a neuron, they're not neuron. He said, what is the definition of the neuron? I challenge you to find the definition of a neuron. So I went looking. Yeah, it's a great question.
Yeah.
I went looking, and I couldn't find anything. And what's worse, there's not an agreed upon definition. What's worse is that there is an agreed upon definition of a neurotransmitter.
Okay.
Which is that basically it should be released from a neuron and some other criteria. But we are lacking one aspect of this, which is, well, what defines a neuron? Well, we don't have an agreed upon definition of that. So,
that's funny.
Yeah.
Okay. So we got somebody asked a follow-up question, or I guess a question that goes along the lines of this discussion. So functionally, though, do these not actually neuron cells conduct signal, though?
I don't believe there is any reason. I don't think anyone has put GCamp in them and shown that they do conduct GCamp. They might have calcium signals within them. A lot of cells have calcium signals with them, right? Muscles as well. But I don't think they conduct signal here. I'm going to tell you this. This is what makes the worm weird.
All right.
The worm has some worm neurons spike. And how many of them spike is still unclear, but they don't use calcium channels for that, I think. They use sodium channels. I'm going to confuse this now because I'm too tired. But they don't have the standard set of channels that basically lead to spiking. Okay. And so I think I've reversed this here, which is basically the sodium channels cause the spiking, and they use calcium channels here to cause the spiking that takes place. And so, and you'll pardon me if I've confused that because I'm a little tired at this point.
You'll be all right.
But worm neurons are thought to be so thin and so small that they're isotonic, in the sense that if you have activity in one portion of the worm neuron, you probably have it elsewhere too.
Okay.
But the fact is, it's not always the case, because from some really nice work from Michael Hendricks, we know that they can compartmentalize their calcium signal. So once you start to look a little bit too much, things get weird. I doubt this is a worm-specific thing. It's just that, there's a lot more neurons to look at for weirdness.
Sure. Once
you get to other animals. But these are hard questions to answer, is a good way to put it.
All right. No, that's the best kind of science. Let's see. In the positional variability map, where is the reference frame centered? Is there a cell that's used as a landmark to determine the positional variability of the other ones?
That's a great question. Yeah. So the way the atlas works is we essentially forge a template through a variety of point clouds that we are basically using an affine transformation. So linear, we can, let's put it this way. If we have 10 worms and we're trying to kind of find a best fit of their best alignment so that as many of these neurons which are labeled sit on top of each other, right? So we try to find a global alignment across all these that works as best possible. Now, we are allowed to deform the different heads using just an affine transformation, which means we can shrink and make them larger. We can rotate them, and I think we can even cause a little bit of shear, but we can't do anything beyond that. So we can't warp locally. And once we start to, you could say, okay, well, why not do local warping? But then, all bets are kind of off, right?
Sure.
Yeah.
Okay, makes sense. How long does the coloring take? I think you kind of answered this, but I think this was a question I had. So I want to be sure.
So it depends what you mean by that. If I interpret that question as how long did it take me to figure out how to color these worms to get that barcode, that was the biggest part of this project, in the sense that I thought I could do it with just with 4 reporters. Then I was like, I need more reporters. So I brought in more. By the time I reached 20 reporters, I'm like, oh, I'm running out of reporters to work with. So then I went through all these papers looking at pictures. I was like, oh, I think that reporter hits what I need. So I tested them all out, went through all the literature. I even, in my desperation, went through some papers and was like, this phenotype looks like it means that this neuron must be expressing that particular gene. Let's take that gene and see if it's in that neuron. And it worked.
Nice.
So, but that was probably the bulk of it. That's, probably, let's see, I started this work in 2014 and I published this, and I was ready with the paper in 2019, which just goes to show you how long things kind of sit in review and then require, reviewer led experiments. Although, yeah, exactly. We had great reviewers, though. I have to say, good. In the reason for the FlyPal design in the paper and the script, basically to bring this to any other animal, all that is due to some fantastic reviewers and clarifying a lot of concepts as well. So I thank the reviewers in real life too, not just as a formality. In terms of the colors emerging, if that's the question, right? At what point in time do the colors emerge? They are, interestingly enough, at around in the embryo, but in the late stages of the embryo, this stage we call threefold embryo, in which the worm starts to move. So while we would love to use these in the embryo, sadly, by the time they're on, this thing is caking. It's moving so fast. Now, all that said, there's a group, Hari Shroff's group, and they work with Daniel Colon Ramos's group, and they have produced some really nice work in basically getting that worm that's twitching and getting good pictures of it, and getting pictures where you can actually see what's taking place. And so potentially we might be able to go one day, not me, somebody else, because I don't want to do that work. It's just so hard. And basically figure out what all the neurons are doing in that embryo, at that late stage where it's, everything's firing and the nervous system is really coming online, so to speak.
That's very cool. All right, good. Let's see. So you touched on this one a little bit, but I'll go ahead and ask it because I think it's something interesting to expound on. Would this technique work in animals that aren't very flat? So like you have cells at different depths that might be different shades of the same color. Would that start to muddle your IDs?
That's a great question. So once, we have some limitations to some extent in terms of the microscopy that we need to bring in, right? So let's say we go to fly, there are ways to fillet the animal, there are slices, but let's say, we don't want to go to that level, right? We have the problem with single photon confocal is that the depth isn't that great, and there's a lot of refraction, and so we would have to bring in these multi-photon techniques to get deeper, right? The problem with multi-photon is, and this gets into kind of more detail that's going to confuse people, and I apologize, I'm going to try to get through it, is easy as possible. The reason these colors are distinguishable from each other is because we have a tuple, which is the excitation laser and the filter, or the excitation wavelengths, I should say, and the filter for the emission, right? And so the reason we can tell blue apart from green is even though this 405 laser even excites a GFP, when we turn it on, we're only looking at what's passing through the blue filter, right? Not through the green filter.
Okay.
And the problem with two photon and multi-photon, the theory, although I've never tested it, and sometimes you find that the theory doesn't hold that much. You can still work a lot with, it still works, maybe, but I don't know in this case, is that it smears it broadens the excitation spectra. And now what'll happen is we'll also, in the case of, I'm relying, you'll note that there's two reds that come through this red filter, right? One is TAG-RP and the other SIOP. But 488 turns on the SIOP and not the TAG-RP, and 561 turns on the TAG-RP but not the SIOP. We're relying on these two different lasers to give us the two different reds separately.
Gotcha. Okay.
But if they smear, we'll run into that problem. Now, all that said, there is a really nice technique which I've wanted to try out from Rohi Yasuda, who I don't know where he's at now. I think he's at Max Planck in Florida. I talked to him, which is called FLIM, Fluorescence Lifetime Imaging. And now, instead of relying on these filters, you excite, and what you really care about now is the lifetime of the fluorescence in nanoseconds. The beauty here is that you can use multi-photon techniques. You can distinguish many different types of fluorescent proteins based off of their fluorescence lifetime, so how long it takes for them to, and you can dark scan, which means that you don't even have to use that much light to get out that signature. Now, all that said, I don't know of too many commercial film systems that can do multiplex a lot of colors. And I was told specifically by the experts that you do homebrew, but you can do it, but it takes a little bit. So, if I get a film expert, I'd love to try it.
There you go.
But for now, that's a dream.
It's always good to have them.
Yeah, but if you have it, you can go deep. You can go deep into cortical areas, and get in there and then read the signature.
Okay. All right. Well, so it's good to know that at least in theory, it's a potential thing that can be done, even if it's technically difficult.
Yeah.
All right. Let's see. So this was a question about the structure connectome, that chart you showed where there was no correlation between the chemical one. So why does structure not determine functional connectome? Isn't the structure the quote unquote mechanism for how neurons can correlate at all? That seems very surprising.
Yeah, I gotta say these are really good questions.
They are.
I don't want to offend anyone in my other talks, but these are a little bit. These have been pretty. These are. I don't want to say it, but these are really good questions. I would say by far some of the best questions I've got.
Well, good. I'm glad.
Yeah, this is great. So there's a lot here that's probably going on. First and foremost is this beautiful work from Eve Marder that I think a lot of people are familiar with about neuromodulation, right? There's state that's taking place. And Eve Marder had shown that once you bring in some of these neuromodulators, like I think it was dopamine and serotonin, and oh man, I'm going to show my ignorance now. I'm tired, but essentially you can create very different neuronal firing patterns in neurons. You have the same neuron, but you basically get a very different set of activity in it, right? And that means that there's some nonlinear function within the neuron. And here, the truth is that we're approximating the correlation through, it's just slightly out of linearity. Meaning specifically that I'm doing, correlation. I also take a log scaling of the synapses. So I say, maybe 100 synapses shouldn't be compared to 10 synapses as like a 10-fold increase. Maybe I should log scale it and it's one order of magnitude, right? And even then we don't see the correlation. So there's partially that. There's partially the fact that in the worm, neuropeptides, and it's probably people are going to kill me for this, but it's probably going to be found to be the case in other nervous systems as well. Neuropeptides we know about signal outside of the synapse. They can have long range effects, and in the worm, we have about 200 types of I think it's 200 types of receptor and 200 different types of neuropeptides, and there are God knows how many isoforms of those, and they have promiscuous binding too. So at certain levels, they'll bind a different type of receptor, and we don't know how they connect to each other. So we've got almost the same order of neuropeptides as we have neurons, right?
Yeah.
And we don't really have access to how they're communicating, so we don't know the graph structure there. There's that. Then there's dopamine and serotonin, octopamine and tyramine, the monoamines, which have all been shown to signal outside of synapses, making our life kind of hard.
Yep.
Then there's potentially the GABA that I showed, and maybe even some of the other neurotransmitters as well, probably potentially leaking out of the synapse and signaling more long range. And then there are states and all of this, and there's starvation. When the worm is hungry, it acts very differently than when it's satiated.
Makes
and
sense.
when it's feeling and there's wanting to have sex and potentially something else, overcoming that urge. And then there's sleepiness and non-sleepiness and roaming and dwelling. Those are the things that we know about.
Yeah.
There's probably a lot more that we don't know about, and we don't have access to state. And I've tried to control these worms as best I can, but their life histories are going to be different, even though their genetics are largely the same. There's just a variety of other things under the hood. Now, all that said, I'm going to tell you about the limitations of the system.
All right.
We record from the soma, the nucleus, basically, and it has been shown that nuclear activity is a good proxy for what's taking place elsewhere. But it's quite possible that there are local computations that we don't have access to, and those, if we had access to them, would marry the two views better.
Okay.
There's also the fact that we're looking at calcium, and that all works on one time scale, but ultimately we're not looking at voltage. So there's that taking place. There's we've assumed some non-directionality for the gap junctions, but they might have directionality. There might be a variability between different worms in terms of their synapses, although that actually has been done. And by and large, while there's some variability in some synapses, the connectome is fairly accurate across these animals, although, maybe it partially accounts for it. And then lastly, I should state that we're recording here at 4 hertz, and the nice thing, of course, about GCamp is that it smears, a fast signal into a signal that can be captured at 4 hertz necessarily, right?
Okay.
So even though you might have a spike that happened on the order of 10 milliseconds, with a GCamp, you're going to have the calcium representation. The waveform isn't going to be a direct spike; it's going to be smeared. Nevertheless, these things are happening a little bit faster than we're recording at.
Sure.
And so, anyone who's worked with GCamp knows that in other animals and vertebrates where you have firing patterns, you get this ramp basically that takes place. It all gets smeared into a kind of like more smooth shape. And all of that is going to basically confound this. Nevertheless, we should have found a better correlation than almost zero. And so there's something there's something more at play here. And there's a lot of factors, and teasing them apart is going to take some work.
Okay.
Yeah, I think that's
cool. Let's see. So I think this will be the last question, unless any more come in. But it's another, I think, interesting one as well. So this relates to NeuroPAL. So you described choosing 3 intensity levels because it makes it easy for human identification. But if a computer system had better intensity resolution, have more IDs you could use, thinking, like a 255 by 255 by 255 RGB space.
Yeah, that's a great question. The answer is yes. And 100% yes. So a computer should be able to do a lot better job than a human being here.
Mm-hmm.
But, and here's the but, ultimately, we have a microscope, and it is, in the case of the original work, it was a oil-based lens for a lot of the stuff, and at 60x, and we have this drop-off of signal, right? And so the further out you go, the less light you get, right? So you can imagine that, the further from the lens you are, the weaker the neurons are, right? And so you would have to have some model that accounts for that and fixes the anisotropy that takes place so that you always have a very stable view of the neuron. Then, in addition to that, there might be some biological noise in the system. And that too, at the level, we don't see that biological noise necessarily at the level of the neuron coloring, when we approximate it as bright, medium, and weak, but ultimately, it might be present at the more finer levels, right?
Okay.
And so, but nevertheless, I should say that a computer should still be able to do a much better job, because we can take many representations of the neuron, build a model, of course, for what it should have looked like based off of the fact that we lost some light with the microscope out, and account for the amount of variance that we normally see for different types at different ages, so to speak. And then the computer should be able to do a much better job of pulling all that apart. And all that said, the panorama reporter, which is in white, it was built by me. I didn't get into this, but the panorama reporters that we had before are all unevenly expressed in neurons. For whatever reason, in the sensory equipment, they were all weak. And so what I did, I tried this also with regular reporters. I'll explain that in a second. But what I did is I took 4 panorama reporters, short regions, I should say, from panorama reporters that we knew from previous work drive panorama expression. So 4 different genes, and I just stitched them together right next to each other and created what I called an ultra-panoronal reporter, UPN. And it worked. And it gave us really even keel, or as good as it gets, expression of that 4th color across neurons. All that said, some neurons are going to be brighter and some are weaker. And that has some information which we never really utilized, I hate using the word utilized. We never really used. We should always avoid jargon when possible. Or just, bigger word. We should avoid the bigger words.
It's hard to break that habit, that's for sure.
Yeah. So we never really used that 4th channel, except for just saying this is a neuron because it's got the 4th channel in it, and this isn't a neuron because it doesn't have it in it, right? And so we could have used it. And so from that perspective, we could use more information in a variety of places. We can use shape. And in fact, some nuclei are big and some are smaller, and that helps sometimes ID. Some are oblong and some are more circular. There's a lot of stuff that we could all kind of use. And even at the level of when you get really good resolution from the confocal, the nuclei look different. And that's probably a result of some of the chromatin. So not all the colors are just expressed in the nucleus, like by driving nuclear expression. Some are attached to histones because otherwise they aggregate. It's an unfortunate thing with some of the colors, basically, that essentially the groups that worked on them did a fantastic job in trying to make them monomeric, but ultimately that meant trying to erase some binding sites that made them either tetramers or dimers, and they still have a little bit of affinity for each other. And the way we solve that, so that instead of you getting a cottage cheese pattern or some weird kind of looking pattern, in order to get the ball-like pattern, I attached it to histones, some of them, not all of them. And that has a very distinct look to it in different neurons. And even that, when you have enough resolution, can help. So that's I think a good illustration of it.
That's an interesting. That's a good point. There's a lot of different ways that you could get at it for sure. Very cool. So I think that's all the questions that we have. So I will say thank you again, Ev, for a fascinating talk. I learned a ton tonight. I don't know how else to say it. I learned a whole lot. This was fantastic. And
thank you so much.
Yeah, go ahead.
Oh, I was just going to say thank you so much for having me, and thank you everyone for listening and for your really good questions. And these are questions that often kind of force me to also think later on and be like, how do we answer that question, right? I mean, it's a great question. How do how do we get at the heart of that? Like, can we maybe carve a little bit of a way so that we get closer and closer to that answer? So sorry, you should go ahead.
Oh no, I was just going to say, you could tell from the questions that were asked that you clearly generated some good thinking as well. So I think that speaks to the quality of the talk.
Thank you.
Yeah,
Thank you, everyone.
absolutely. So thank you again to everybody for coming out. We will be back next week. Hopefully, with another great talk. So I hope everyone who's out tonight can come back again next week. Thank you again, Ev. If I can get you to hang around for just 1 second, we'll talk very briefly. So we'll see everybody next week. Have yourself a good weekend. And stay safe. Good night, everyone.